Today we'll discuss photosynthesis, some question of photosynthesis. So previous year question like Aims question or Neet question. So first is that correct sequence of cell organelle during photorespiration. During photorespiration, we know that during photorespiration, there's three organelles involved. That is called I told you to remember in that way CPM. CPM is full form is chloroplast, peroxisome, and mitochondria. These three organelles are involved. Now see that option what's actually given. This photorespiration, also known as C2 cycle or respiratory pathway. It's a three organelles. See that first chloroplast, Golgi board. No, Golgi board is not involved. So option is rough endoplasmic reticulum. No. Now see that vacuole. No. So option 3 is the correct answer. Chloroplast, peroxisome, and mitochondria. Right. Now next question. The total requirement of ATP and NADH from one molecules of total requirement of ATP and NADH for each molecules of carbon dioxide fixed and reduced in photosynthesis in the Kelvin cycle. So Kelvin cycle, we know that during Kelvin cycle there is a first is that if for 6 CO2 fixation, how many molecules of ATP is required? 18 molecules of ATP and 12 molecules of NADH required, right? So for one molecule of carbon dioxide fixation, one molecule of carbon dioxide fixation, it will be 3 ATP plus 2 NADH molecule. NADPH molecule is required, right? So answer will be 3 ATP and 2 NADH. Option 3 is the correct answer. 3 ATP and 2 NADH. Because you know that during 6 RUBP plus 6 CO2 form 12 PGA. 12 PGA to 12 PGA ALD formation. That step required 12 molecules of ATP. Another is the during regeneration phase of RUBP, 6 ATP is required. So 12 ATP molecules, and another is that 12 PGA phosphoglyceric acid to phosphoglyceraldehyde formation. There are also 12 NADPH is used. This ATP and NADPH actually synthesized during the light phase of the photosynthesis, right? That's why dark reaction or Kelvin cycle is not independent. Kelvin cycle dependent on light reaction on, like in that case, in energy respect, right? Now next question, a process that makes the important difference between C3 and C4 plant. C3 and C4 plant, a major uh, difference, in respiration and say transpiration. No, transpiration is not. You all know that in case of C4 pathway, there is a major changes occurs in C4 pathway is that in terms of photorespiration. Photorespiration to remove because photorespiration is possible because of Rubisco enzyme. Rubisco is present in the in case of C3 plant in the mesophyll cell. Now in case of C4 plant, it is transferred into bundle seed cell to reduce the respiration. As the photorespiration is possible, Rubisco able to come into contact with oxygen. So that's why Rubisco is shifted into the new compartment, that is say bundle seed cell, right? So difference in case of photorespiration. So next question. Now See that cyclic photophosphorylation, cyclic photophosphorylation results in the formation of cyclic photophosphorylation. We know that during cyclic photophosphorylation, during cyclic photophosphorylation, what's actually happened during cyclic photophosphorylation? There is two type of photophosphorylation system we have seen, that is non-cyclic and cyclic photophosphorylation. I have told you during non-cyclic photophosphorylation, non-cyclic non-cyclic photophosphorylation, non-cyclic phosphorylation synthesis, ATP and NADH both actually synthesize non-cyclic. ATP and NADPH both actually synthesize during non-cyclic. But in case of cyclic photophosphorylation, what happens? Cyclic only during cyclic photophosphorylation. Sure. See you later. Now coming to that, first say এখানে non-cyclic photophosphorylation ATP and NADH produce. In case of cyclic, I have told you during cyclic photophosphorylation only ATP is produced. So answer will be only ATP produced. So option C is the correct answer. So next question. So the enzymes catalyze the carbon dioxide fixation in C4 plant. Which enzyme actually during C4 plant? I have told you there is a the CO2 acceptor. In case of C3 plant, CO2 acceptor is RUBP, ribulose 1,5-bisphosphate. RUBP actually the CO2 acceptor. RUBP combined with CO2, RUBP combined with CO2 to form PGA or phosphoglyceric acid, right? So phosphoglyceric acid is in case of C3 plant, but in case of C4 plant, that is a PAPE, that is phosphoenol pyruvate, actually acts as a CO2 acceptor, right? And it produces oxaloacetic acid. Phosphoenol pyruvate plus CO2 produce oxaloacetic acid, OA, C4 pathway, right? So here the enzyme is PAPE carboxylase is the major enzyme. So major enzyme is PAPE carboxylase, major enzyme. So next question. In Cranch anatomy, the bundle seed cell have many intracellular space. In case of Cranch anatomy, what we have seen during Cranch anatomy, there is a special arrangement occurs. So Cranch anatomy is the bundle seed cells have during branch. So you can see that this arrangement of Cranch anatomy. This is ultra structure of the leaf. Now see that there is a these are mesophyll cells. And now these are bundle seed cells. So bundle seed cells with large chloroplast and mesophyll cells with few and small chloroplast. Actually, this type of arrangement, like see that bundle seed cells and mesophyll cells is outside. These are mesophyll cells in the outside, and bundle seed cells is the inside. And inside the vascular bundle is located. So this type of arrangement is called Cranch anatomy. So Cranch anatomy, the bundle seed cell actually say thick wall, bundle seed cell is thick wall, no intracellular space, and large number of chloroplast. Thick wall, no intracellular space, and large number of chloroplast. Right? So answer will be option B. Now next question. C4 pathway is advantageous over C3 pathway in plants because in case of C4 pathway, we know that C4 pathway actually there is a in C4 the carbon fixation actually done by a PEP carboxylase enzyme. Enzyme is PEP carboxyl. In case of C3, there is a Rubisco enzyme is there, but in case of C4 pathway, it's if it is a low CO2 concentration, so it can be able to photosynthesize. So C4 C4 pathway C4 pathway is saturated usually saturated at relatively low CO2 concentration. C4 plant C4 plant has greater photosynthetic yield in low CO2 concentration. So answer will be option A. That is occurs relatively low CO2 concentration. Next, the first carbon dioxide fixation product in C4 plant. I have already told the first carbon dioxide fixation product. First fixation product is oxaloacetic acid is the first fixative product. PEP plus CO2 together it form OA or oxaloacetic acid, right? So answer will be option B. Next. Now the match the column. Match the column here different name of scientists given. So first is that Stephen Hells. It's actually first is air and light are controlled plant growth, and B actually will be A, that is in importance of light and chlorophyll. C is the von mole. C actually the presence of chlorophyll in plants, and D sac actually 3 product of photosynthesis is a starch that is discovered by sac. So answer will be option A. It's the correct answer. Now next question. During photosynthesis, oxygen in glucose comes from. Oh, okay. So I have told you that there is an experiment, radioactive experiment is done. So 6 CO2 plus 12 H2O, after reaction it will form C6H12O6 plus 6O2 plus 6H2, right? So now the question is asking that during photosynthesis oxygen in glucose during photosynthesis oxygen in glucose comes from. Now glucose actually glucose, you all know that the oxygen actually coming from H2O. This is actually radioactive leveling experiment. I have told you that radioactive leveling is proved that the oxygen during photosynthesis coming from water, and the glucose oxygen, this glucose oxygen is coming from carbon dioxide, because carbon dioxide also contain oxygen. This oxygen actually coming into glucose, and the H2O oxygen coming into that in the oxygen in as a oxygen releasing form, right? So finished. ওকে, চলো স্টপ করি।